Today we're going to talk about gene mapping using the example of the Brocade and Merit corn, where Brocade has a lazy phenotype that's conferred by a homozygous recessive genotype, and Merit has an upright phenotype, so they have a capital LZ gene. As we go forward with this problem, you might want to get out your recombination practice worksheet to follow along, because I'll be referencing that as we go through. So, what we know about this lazy mutation is that it's somewhere on the chromosome, but we don't know where yet. So let's look at what we do know about the Brocade and Merit chromosomes. So here are our two chromosomes. And I'll call this one the Brocade parent, and that one the Merit parent. Now, we know already that there are some genetic differences between Brocade and Merit. So for example, we know there's a region up here on the end of the chromosome that I'm going to call region 1 that's got a different number of base pairs between Brocade and Merit. We can use that as a PCR marker. Since they're different in size, we can tell the difference in the chromosome region. We've got another marker down here. I'm going to call that region 2 or marker 2. And since, again, marker 2 has a different number of base pairs between Brocade and Merit, we can use a PCR and then look at a gel electrophoresis to tell which version of the chromosome we're looking at. Are we looking at the Brocade version or the Merit version? Now, on these chromosomes, there's other pieces of information that we already know. We already know that there are two other markers on these chromosomes, marker 3 and marker 4, and again, they're different between the Brocade and Merit parents. It's important that we know this information already exists, because in order to map the LZ mutation, we need to know something about what the chromosomes look like already. So in the story we're working on, we want to map the LZ mutation, so we've crossed Brocade and Merit together to create an F1 generation of plants. These are going to be heterozygous for the lazy mutation. And then if we cross the F1s together, if we self them, we're going to create an F2 population that's going to be segregating. Some of the F2 plants will be homozygous for the wild type upright phenotype. Some will be heterozygous, and some will be homozygous for the lazy mutation. Now, as we figure out where this lazy mutation is located, we want to consider only these F2 plants that have the lazy phenotype. So we're only going to be considering these homozygous plants. And what we want to ask is this lazy phenotype, or really is the little LZ allele associated with one of these markers found on the chromosome? Can we identify which marker or markers is most closely linked to LZ? Linked to marker, we don't know which marker yet. So let's talk about the data that we have, the gel data that we have on our worksheet. First of all, we're considering marker 1. And remember, we're looking only at the LZ homozygous F2 plants. Now, if you have inherited a chromosome that has the LZ mutation on it somewhere, it's also going to have it's going to have all 4 of these other genetic regions or other markers. And we want to ask, for marker 1, does little LZ tend to stay linked with the Brocade version of that marker, or does it also associate with the Merit version of that marker? So by counting things out, we're looking at 20 different plants. Since each plant is diploid, they're going to have 40 chromosomes. So all 40 of these chromosomes are carrying the little LZ genotype. And if we count things out, we'll see that 20 of the chromosomes have the Brocade version of marker 1. That's out of a possible 40 alleles. And 20 of the alleles are the merit allele out of a possible 40. So 50% of the time, little LZ is sticking with Brocade, and 50% of the time, little LZ is associating with merit. Since that's about 50-50, that's like exactly 50-50, that means that LZ is not linked to marker 1. It can freely or independently associate with either version of marker 1, regardless of what parent LZ originated from. And we know it originated from the Brocade parent. So now let's contrast that to marker 2. In marker 2, again, we're looking at only the LZ homozygous F2 plants. And by counting up the alleles in the gel, we can see that 32 out of the 40 alleles we're looking at have the Brocade allele, and 8 out of 40 have the merit allele. So if we do the math, that's 20% have the merit allele, and 80% have the Brocade allele. So the LZ allele, wherever it might be located, stays with the Brocade allele of marker 2 80% of the time, and only 20% of the time do we see the little LZ allele associate with merit. So what we can do here is draw the LZ allele on the chromosome. We know now that LZ must be linked, closely associated with this marker 2. So I'm going to draw LZ next to it here. So I'm drawing the little LZ on the Brocade chromosome and the wild type LZ on the merit chromosome. And since I know that 20% of the time we actually see this LZ allele with the merit version of the chromosome, we know that 20% of the time there's recombination between that location, those two locations. So 20% of the time we see recombination here. That helps us know that this distance between marker 2 and LZ is 20 centimorgans. So 20% recombination, 20 centimorgans. Now, a next question we might want to ask is, what's the genotype at marker 4? Do we see that LZ is also linked to marker 4? I would predict that LZ is less closely linked to marker 4, given how we've drawn it here. It's a little further away. But we'd have to do the genetic testing and ask for marker 4, what proportion of LZ mutants have Brocade, and what percent of LZ mutants have the merit allele at marker 4. And that's going to tell us how far this distance is between marker 4 and the LZ allele. Now, altogether, by looking at the different marker loci on these chromosomes that we already know exist, this is prior knowledge that we have to have in order to map where these genes are located. It's only by using this previously existing knowledge about markers that we can map our gene of interest, the LZ gene.